Erev Tov, good evening.
Good evening.
Hi. We'll wait for a few more minutes. Everybody join in. Good evening again. One more minute and we'll start. Okay, let's start. Let me share my screen. So let's start with asking, how are you doing this evening?
Pretty well. How are you?
Sorry?
I'm doing pretty well. How are you doing?
Very good, but I want you to use Mentimeter.
Oh. I just joined this class, so I don't know exactly what is a Mentimeter.
So look at the top of the slide. Take your phone out and follow the instructions. You go to this website, put in this code, and type in whatever the question is. So this class is about participation. There will be other questions, and I do hope that you participate. And Khaled, before I forget, if you can take attendance, please. Okay. All right, so this week overnight, we went from 15 to almost 30 students in this class, and I suspect this is because the class was not advertised broadly beyond physics department, and now students from other departments have joined as well. It's a little bit of an issue because all the new students missed the first two meetings. I'm actually giving lectures and speaking only for 6 of the meetings we are going to have. We are going to have 13 meetings altogether during the semester. And I'm presenting during 6 of them. And over the second part of the course, for 7 meetings, we will have you actually present to everybody else. So first of all, everybody has to prepare a PowerPoint and a PDF as is listed in all the instruction I gave already. And the attendance is very, very important because I want everybody to be there when students speak. So we'll take attendance. And we'll have to do some adjustments because we are going to have way more students than what I anticipated. So the length of the presentation will shrink. We are going to accommodate 4 students per meeting, 20 minutes each. So like 7 minutes presentation plus 3 minutes for questions and discussions. We might adjust this a little bit further as we go down and see if everybody that is listed right now to the course is participating, or there are some people who are going to leave us. But basically, you should plan and you should start to look for topics to present. I already had one student communicating with me, and I authorized his choice. So this is about the administration comments that I had. Anybody has questions, requests, complaints so far? If you do, please turn on your mic. Okay, if nothing else, as we discussed at the beginning of the course, the topics that we are raising here are current. There is an ongoing discussion in all kinds of social media and in scientific literature. So here is just one example for March of this year about open science. And this is a view that open science is good, but it can also make big problems. So I recommend you guys, if you're into read. We are going to talk about misconduct, plagiarism, and all kinds of things of that sort. And this is again for March of this year. This is Vice Chancellor for Research in North Carolina. And he caught plagiarizing. So, again, we think that science is pure and everybody is really very honest and doing the right thing, and we implement the scientific method the best way we can. But you will find out during this course that this is really not the case. And there are many cases of misconduct, and peer review usually doesn't catch those. So that will be a big topic for discussion. And obviously, I prefer that when you pick up a topic to discuss, it's either a misconduct or a big error, mistake, retraction, things of that sort. This is from a few days ago. This is November 3rd of this year. We talked about how all the scientific journals responded very quickly to the pandemic, and all research related to COVID was immediately made public, even by journals that require subscription. That was a very, very nice thing to do. Obviously, the idea was to help coming up with vaccines and therapeutics as fast as possible, and subscription walls don't help in that regard. So all journals really responded properly for this pandemic. It actually turns out that they've done it not only for COVID, they've done it for Zika, for Ebola, and for monkeypox as well. But there are some discussions already, and you can read this commentary. This is from Nature of last week. COVID research is free to access, but for how long? Because there are some discussions that are going to put again, the subscription walls on these papers after 2 years or 3 years of pandemic. So again, it's an interesting read about the response to the pandemic, and then for how long are they going to do this? They're losing money. Okay, so I asked all of you to read 2 papers for today's class. So let's just ask if you did that. I ask everybody to respond because I want to have an idea. I guess that the new students did not know about this assignment. Still, we have 24 people on the Zoom and only 18 answered. Okay, let's move on. So we are going to cover today two examples for very, very good and important papers. The first one is Watson and Crick on the structure of double helix DNA, and the paper by Forster on energy transfer. This will also be a segue for my own research, and we are going to discuss a little bit next week about my own research. So, in fact, the paper by Watson and Crick is not a single paper. There were 3 papers published back to back, and here are these papers. But before we do this, let's ask you another question. Since there are 3 papers, but only one is very well known, I would like to ask you, why is Watson and Crick paper so well known? And as a result of that question, why the other 2 papers are not as well known? So at least people who read the paper should be able to answer. Molecular structure of DNA. It's a very fundamental result that impacts us even today. They were correct, very well written. Huge discovery. Guess what? It's a huge discovery, and correct results show up in the other 2 papers as well. Led to Nobel Prize. They were spot on. Published in Nature. Plagiarism. I'm not sure that's true. Whoever wrote plagiarism, if you can turn on your mic and explain why you think there is plagiarism there.
I said it. So basically, they copied another research work. So it started that it was very publicized because it was the first one that published the DNA. But later on, and we're talking like 2004, it was mainly known for the plagiarism after they received the Nobel Prize.
I think you're using the wrong word. It's not plagiarism. They used data without permission. We'll talk about this. But they did not copy somebody else's work.
Well, they sort of did. Like they took something that wasn't theirs.
No, we'll talk about this. That's not exactly the case. But it definitely doesn't fall into plagiarism. We'll discuss what plagiarism is. There was a misconduct, most likely. But it's not plagiarism. So we'll discuss what it is. Any other ideas, comments? All right, so let's start. Okay, so here the first paper. This is Watson and Crick. It starts here and it ends here. This is 1 page in Nature. 1 drawing by hand, no data. So that's the right citation. This is this paper. And then you see this figure, this is data, but this is not their paper. This is a second paper. Oops. So that, yeah, that was the first paper. Here is the second paper. This is by Franklin and Gosling. And in this paper, there is some data and a single equation. And instead of one page, it's 2 pages. And the third paper is, wait a minute, something, okay, yeah. We have Franklin and Gosling, this is this one, and we have Wilkins and, okay, I messed up. Okay, so here's the first paper. This is Watson and Crick, 1 page. The next one is Wilkins, Stokes, and Wilson. And this is 2 pages and a bit. And this is the third paper. This is Franklin and Gosling. Okay, so the data that Watson and Crick rely on actually shows up in this paper, in Wilkins, Stokes, and Wilson. And those 3 papers are back to back. They are all in the same issue. They are consecutive, one after the other. There was an agreement between the authors of all 3 papers and the editor, and they all decided to put those 3 papers together. We know mostly about 1 paper. So we asked you that question already. And what I'm going to do now, I'm not going to analyze the paper myself. I'm actually going to rely on an analysis that used to be displayed in a science museum called the Exploratorium, which is in San Francisco. It's a beautiful hands-on science museum. When I was in the Bay Area, I was there for 10 years, from 1990 until 2001. My kids were young at the time, and we used to go to that museum often because it was really a very, very good hands-on museum for kids where they could try and learn and do experiments. And so I don't know if this display on double-stranded DNA is still there or not, but what we are going to do next is we are going to analyze the paper as it was displayed in that museum. So here's the paper. Just minimize this zoom bar. That's no, that's not good. Okay. So here is the paper. This is basically the text of the paper. And what we're going to do, there are comments in this display in the museum, and we are going to look at those comments. So there is a comment about the authors, about the date, about the journal it was published in, reference to polling that Watson and Crick are referring to in their introduction. So let's start to go through this. So basically, this is a long paragraph about the authors themselves. I'm not going to go and read everything here, just to say that Watson and Crick were on this problem for quite some time. Pauling was really leading the area of macromolecular structure using X-ray. He solved the alpha helix of a protein 2 years before. And Watson and Crick knew that he's after the structure of DNA. And it was clear that DNA is the genetic material and the secrets of life, and how genetic information is being propagated is happening through DNA. So there was a huge incentive to really understand and solve the structure of DNA. Watson was a postdoc in Crick's lab. He actually did his PhD in the US with Szilard. Szilard was an Hungarian Jewish scientist who escaped the Nazis and joined the Manhattan Project. And after the Manhattan Project, there were quite a few physicists interested in the secret of life. They were very much influenced by the book by Schrödinger. Actually, let me show this to you. Just a second. So what I'm going to show you in the camera is going to be a mirror image, so it's not great, but can you see this? Actually, I need to stop the background just a minute. Let me stop the backgrounds. It'll take me a second. Okay, yeah, I have to do this. Come on. I think I have to stop share just a minute, okay. Okay. So as I told you, a huge influence on the field of molecular biology, of biology in general, is this book by Schrödinger, who wrote it in, I think, the early 40s, What is Life? And lots of very interesting ideas and questions come. You know, when you get your Nobel Prize in quantum mechanics, you have nothing left to do. You basically start to think about big questions. And he's not the first Nobel Prize to do that. There are several Nobel Prizes who went into all kinds of philosophical questions. Schrödinger actually asked very, very good questions about biology. So he really influenced lots of physicists. And after the Manhattan Project, when the war was over, there was this phage club at Cold Spring Harbor that was assembled several different physicists to try to understand life. And they asked, what is the simplest form of life? And they started to do all kinds of interesting experiments. Szilard was one of them. And Watson was his student, PhD student. And when he finished, he moved to the best place in that time to do crystallography and salt structures, which was in the MRC lab in Cambridge in the UK. So this is how he joined the Crick lab. All right, let's go back to the presentation. So that, actually, I wanted to show you that book. I also want to show you, we'll talk about this later, but there is this book as well. This is a book by Watson about the double helix. The name of the book is The Double Helix. I highly recommend those of these books for all of you to read. Now let me share again. I lost my. Okay. Okay, so Watson joined the group. The group is competing with Pauling. Pauling doesn't really know that they're competing with him, and they are trying to get first to the structure of DNA. There were several key experiments before that really showed that DNA is the important player here. So that's this comment about the authors, and then Nature. Again, I'm not going to read the whole paragraph, but what is really interesting okay, sorry. The comment about the date. So as I said, there is a competition. Pauling is in Caltech in California. Watson and Crick are in Cambridge. Historians wonder how the timing of the DNA race affected the outcome. Science, after years of being diverted to war effort, was able to focus more on problems such as those affecting human health. Yet in the United States, it was threatened by a curb on the free exchange of ideas. Something that American researcher Linus Pauling would have beaten Watson and Crick to the punch if Pauling's ability to travel had not been hampered in 1952. So, anybody has an idea what we are talking about here? What is the House Un-American Activity Committee? Turn on your mic.
He was a pacifist, and at that time there was a movement against communism, pacifism, etc.
What is the name of that movement?
McCarthyism.
McCarthyism, exactly right. Pauling was a pacifist after the war, and he was very loud and active. And because of that, McCarthy put him on the list. He was on the blacklist, and he was not allowed to travel for scientific conferences. And many think that if he had a chance in 1952 to go to a meeting in Europe and see Wilkins and Franklin and talk to them, because he was ahead, obviously with proteins, but he already had some crystals of DNA. He got some diffraction from those crystals. He came up with a model, which is the wrong model. But if he had all the information that Watson and Crick had at that time, he probably would have gotten there first. But he did not go to that conference because he was not allowed to go to the conference. Nature. You all are familiar with Nature. Everybody wants to publish in Nature. That was true since the beginning of that journal, 1869. Hundreds of other scientific journals help push science forward by providing a venue for researchers to publish and debate findings. Today, journals also validate the quality of the research through a rigorous evaluation called peer review. We discussed peer review in the first two meetings. Generally, at least two scientists selected by the journal editor judge the quality and originality of each paper, recommending whether or not it should be published. That's not true for the double helix DNA. Science publishing was a different game when Watson and Crick submitted this paper to Nature. With no formal review process at most journals, editors usually reach their own decision on submissions, seeking advice informally only when they were unfamiliar with the subject. So basically, the editor decided if the paper is accepted or not. The paper has no data. The paper has no figure of results. The paper has one hand drawing of a presumed structure of DNA, was accepted by the editor, and this is how easy it was at that time to publish a paper. Okay? And we discussed already about Pauling, so again, as I said, Pauling was planning, he wanted to go to that meeting in London, but he was refused. And as Yehuda said, yes, he was a pacifist, and what is really, life is always full of surprises. The year that Watson and Crick got their Nobel Prize in Stockholm for the double helix, Pauling got his second Nobel Prize for peace, same year. Very befitting. He missed the race, but he got two Nobel Prizes, and he got one of them at the same year that Watson and Crick got theirs. Okay, so obviously I'm not going to read the paper itself. I'm just going to highlight the points that are being discussed here. So they say we wish to put forward a radically different structure for the salt deoxyribose nucleic acid. This is because Pauling put in writing a proposal for a triple helix before them. So here, the young scientists Watson and Crick called the model radically different to strongly set it apart from the model proposed by Linus Pauling. This claim was justified while Pauling model was triple helix with the base sticking out. The Watson and Crick model was double helix with bases pointing in the forming pairs of A and T and C and G. Okay, so they're talking here about the two strands that are basically opposing each other. The central description of the double helix model still stands today, a monumental feat considering that vast majority of research findings are either rejected or changed over time. Obviously, that initial result of the double helix holds today as good as it was holding in 1953. According to science historian McElhenney from MIT, the staying power of double helix theory puts it in class with Newton's law of motion. Just as Newtonian physics has survived centuries of scientific scrutiny to become the foundation of today's space program, the double helix model has provided the bedrock of several research fields since 1953, including biochemistry of DNA replication, cracking the genetic code, genetic engineering, sequencing of the human genome, and so on. So basically, all molecular biology and biotechnology of today is relying on that one result from that one paper. Furberg basically did some experiments before Watson and Crick, and he actually hinted to the correct structure, and Watson and Crick basically cite him. He correctly put the bases inside the helix. Both polling model and other discussions in the field suggested that the bases are sticking out. Anybody knows how we call a DNA where the bases are sticking out? Any idea?
RNA.
It's called pDNA. P like Paul, pDNA. And this structure actually can be generated in the lab. If you pull on a DNA with magnetic tweezers and you supercoil it a lot, then the bases will basically flip out and you'll have the sugars in the center and the bases facing out the solution. But you have to put a lot of energy and strain into the DNA to get that structure. But initially, people thought that this is the structure of DNA. So the first suggestion that the bases are actually facing each other and they are inside and the strands are outside was by Furberg. And that helped, obviously, Watson and Crick to get to the right structure. So obviously, every terrific scientific result always relies on other pillars that did some work before. And here, Furberg's contribution was very, very important, not acknowledged enough. Figure. Who drew that figure?
Watson's wife, I believe.
Yeah. So the two ribbons symbolize the two phosphate sugar chain, and the horizontal rods, the pairs of bases holding the chain together. So a visual representation of Watson and Crick model was crucial to show how the components of DNA fit together in double helix. So Crick's wife, Odile, drew the diagram used to present DNA in this paper. Scientists use many different kinds of visual representation of DNA. For your generation, you cannot imagine this. But when I was a PhD student at the Technion, we still had people in the office who did the typesetting. So you could basically give handwritten manuscript to somebody and they will type it, not on a personal computer. There were no personal computer. They were doing it on a mainframe with a terminal. And there was 3 or 4 people in the department who were doing drawings. You would sketch something and they will make drawings for you. And that was the role of Crick's wife. She actually just drew this by hand. And this is possibly the most famous scientific drawing ever. So nowadays we all use PowerPoint and all kinds of other tools, and it's very easy. But those days, preparing a manuscript for submission took many, many months. Okay, so here we talk about the base pairing. The last hurdle for Watson and Crick was to figure out how DNA 4 bases paired without distorting the helix. To visualize the answer, Watson built the cardboard cutouts of the bases. And the bases have 2 or 3 rings with 2 or 3 hydrogen bonds. And that was really the big puzzle. So they had the X-ray diffraction. The X-ray diffraction, we talked about this already last time. They knew already from mathematical calculation that the diffraction pattern should be in the shape of X, because that would suggest a double helix structure. But they needed to fit the bases in. And to do that, they basically drew those 2 or 3 rings on a cardboard, cut it out, and started to play with this and build a physical model to play with this. Okay. To visualize the answer, Watson built cardboard cutouts of the bases early one morning. As Watson moved the cutouts around on the tabletop, he found that only one combination of base molecules made a DNA structure without bulges or strains. As Crick put it in his book, I showed you the Watson book. There is another book by Crick, and that book is named What Mad Pursuit. Watson solved the puzzle not by logic, but by serendipity. And oftentimes we get big discoveries just by serendipity. Watson and Crick picked up this model building approach from eminent chemist Linus Pauling, who had successfully used it to discover the helix structure of alpha helix. So basically the model building is a very important tool. Nowadays, you know, you go to chemistry department all over the world and they have this ball and stick kits where you can build all kinds of molecules. That's basically what they did, but they did it with cardboard. Probably impossible. Okay, sorry. So comment 10. Alongside Watson and Crick paper in April 25, in the same issue, we have these other 2 papers that I've shown you, Maurice Wilkins and Rosalind Franklin, who work independently of each other. And Wilkins and Franklin papers describe the X-ray crystallography evidence that helped Watson and Crick devise their structure. The authors of the 3 papers, their lab chiefs, and the editor of Nature agreed that all 3 would be published in the same issue. The following communication that our authors are referring to are the papers of Franklin and Wilkins published on the journal pages immediately after Watson and Crick. So here, so far as we can tell, it is roughly compatible with experimental data, but it must be regarded as unproved until it has been checked against more exact results. Some of these are given in the following communication. We were not aware of the details of the results presented there when we devised our structure. So here comes a big no-no. They basically say they have not seen the data of Franklin before they came up with that structure. And this is simply a blunt lie. So they basically say we were not aware of the details, which rest mainly, though not entirely, on published experimental data and stereochemical arguments. So they say we got to that structure based on already published data and some unpublished data. They don't say what, but they say we have not seen the data of Franklin. So here there is a big comment, and I'm going to read that comment because this comment is very important. This sentence marks what many consider to be an inexcusable failure to give proper credit to Rosalind Franklin, a King College scientist. Watson and Crick are saying here that they were not aware of Franklin's unpublished data, yet Watson later admits in his book, The Double Helix, that these data were critical in solving the problem. Watson and Crick knew this data would be published in the same issue, but they did not formally acknowledge her in the paper. What exactly were these data and how did Watson and Crick gain access to them? While they were busy building the models, Franklin was at work on the DNA puzzle using X-ray crystallography, which involved taking X-ray photographs of DNA samples to infer their structure. So basically, the one person that did all the work was Rosalind Franklin. She was crystallizing many, many DNA crystals, taking each of those crystals to the X-ray machine, collecting data and collecting data and collecting data. This is a person who did the work. So by late February 1953, her analysis of these photos brought her close to the correct DNA model. She was not only collecting data, but she was actually trying to solve the structure. But Franklin was frustrated with an inhospitable environment in King's College, one that pitted her against her colleagues. And in an institution that barred women from the dining room and other social venues, she was denied access to the informal disclosure that is essential to any scientist's work. So basically, she was basically gender identified and basically pushed away. At that time, King's College, I don't know if you know King's College, it's in a very nice part of London. There is a beautiful market next to it. It's a very, very ancient hospital and medical school. In fact, it's such an interesting old place that they are doing archaeological excavations. And about 20 years ago, they found a surgery room from, I don't know, 15th or 16th century on that site. And they have a nice museum there of old, surgery room tools. So if anybody gets to London, go to the museum in King's College. Anyways, she's in that college, and females are not allowed in the dining room. And she cannot have scientific discussions with colleagues in the corridors. And she's very frustrated and she's very upset. And she decides she's doing this very, very important work, but her conditions in that place are so deplorable, she decides to leave. So Franklin decides to take another job. As she was preparing to leave, she turned her X-ray photographs over to her colleague, Maurice Wilkins. She was not a collaborator of Wilkins. She was not working under Wilkins. He was the closest to her field, and they were more or less talking, not obviously in the dining room, but they had some discussion. Because she decided she is leaving, she took all her data and put it on his desk. And Wilkins, on the other hand, was a friend of Crick. Then, perhaps the most pivotal moment in the search for DNA structure, Wilkins showed Watson and those photographs that Rosalind gave him without asking for her permission. It's her data. Her colleague who got that data decides to give it to the competitor without asking for her permission. Wilkins showed Watson one of Franklin's photographs without Franklin's permission. As Watson recalled, the instant I saw the picture, my mouth fell open and my pulse began to race. To Watson, the cross-shaped pattern of spots in the photo meant the DNA had to be double helix. Was it unethical for Wilkins to reveal the photographs? Should Watson and Crick have recognized Franklin for her contribution to this paper? Why didn't they? Would Watson and Crick have been able to make their discovery without Franklin's data? For decades, scientists and historians have wrestled over these issues. I would dare say that if we were today, there was no way they could publish this paper without Rosalind Franklin, an author on that paper. She would go to the editor and object, basically, the paper, and then they couldn't publish. So we have gone quite a bit over the past 70 years, in terms of scientific publishing and what is ethical and what is unethical. But this is an interesting story. It's not plagiarism. It's just using somebody else's data without permission. Now, they add one comment after they basically present the model, and that's a very, very interesting comment. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism of the genetic material. This phrase and the sentence it begins may be one of the biggest understatements in biology. Watson and Crick realized at the time that their work had important scientific implications beyond a pretty structure. In this statement, the authors are saying that the base pairing in DNA, adenine links to thymine and guanine to cytosine, provide the mechanism by which genetic information carried in the double helix can be precisely copied. Knowledge of this copying mechanism started a scientific revolution that would lead to, among others, advances in molecular biology, the ability to manipulate DNA for genetic engineering and medical research, and to decode the human genome, along with those of the mouse, yeast, fruit fly, and others, and all the way to CRISPR-Cas9 and genetic therapy and everything that you hear about today, and coming up with an mRNA vaccine for COVID within 9 months after the pandemic started. So this is all relates to this one discovery. But they say it in a very nice and modest sentence. They were not modest about Rosalind, but they are modest here. It has not escaped our notice. Okay, and we talked about the fact that the other two papers are 2 pages each. This paper is only 1 page. This paper is short because it was intended only to announce Watson and Crick discovery, and because they were in a competitive situation. In January 1954, they published the full details of the work in a longer paper that was in a proceeding of the Royal Society. This expound later approach was usual in science in the 50s, as it continues to be. In fact, Rosalind Franklin did the same thing, supplementing her short paper with another 2 long papers. So there was no internet. The only way to communicate was really by mail, snail mail, surface mail. Even phones were very expensive. International calls were expensive and difficult. So scientists who wanted to talk to each other really exchanges ideas by mail. And you did not know what the competing group is doing. So the philosophy was you have to publish very quickly once you have a result, and then clean up later. So you publish this 1 page, and later on you clean it up, because you want to be first. So that's the end of the paper. Some acknowledgement. Obviously, Rosalind Franklin is not. No, she's acknowledged, but by her data. So just a minute. Let's read what they say here. We are much indebted to Dr. Jerry Donahue for constant advice and criticism, especially in interatomic distances. We have also been stimulated by knowledge of the general nature of the unpublished experimental results and ideas of Wilkins and Franklin and their co-workers at King College. So and by the way, nowadays we like to give credit to our funding agencies because this is how we'll get more money later. And they have done this well over when they publish this paper. So Watson is acknowledging a fellowship he got. But again, look at the language. They are not saying we got the data. We used the data. We were aware of in the neighborhood. All right, so that's the analysis on Watson and Crick paper. I want to refer you, I'm not, again, going through this at all, but obviously this paper goes beyond science. In fact, it became an ethos. And if you're interested, this is a very interesting read by Michael Halloran, The Birth of Molecular Biology, an essay in the rhetorical criticism of scientific disclosure. This paper talks about establishing an ethos, competitiveness between the teams, ethics, gentile style lengths, and so on. So it's an interesting read. I recommend you to read it. It's different. It's not written by a regular scientist, but very, very interesting. And to conclude, this is 50 years after the discovery. Tobin is basically writing an essay, 3 papers, 3 lessons. First lesson, collaborating with faculty colleagues is more profitable than competing against them. So that's really common to Linus Pauling, but Pauling really did not have a chance because he could not travel to the most important meeting of that time. Second lesson, research has no value if unpublished. That really relates to Franklin. She had the data and she gave it away and she moved to another place because she was very, very unhappy with the environment in King's College. And third lesson, Watson and Crick have something important to say. They say it clearly and then shut up. The article exemplifies the three cardinal qualities of good scientific writing. Be brief, be clear, be simple. So that's an attempt to give you an answer why that one paper is much more well-known compared to the other two. All right. Any questions, comments about the double helix? Thoughts? What happened to Franklin, to Rosalind Franklin? Anybody knows? So very shortly after she moves, I don't remember where she moved to. She actually...
She died at the age of 37.
Yeah, she got cancer. So very, very sad story. Nowadays, we all know her contribution and acknowledge, but very, very sad story. Okay. Well, if no more comments about the double helix, let's move to the next question, which is, did you read the Forster paper? And I want to get an idea who read it and who did not, so please answer. So more of you did not read compared to people who read it. Not ideal. This paper is much more difficult to read compared to the double helix paper. I'm not surprised. So this is a paper we want to discuss. In fact, there are two versions. This is a version published in Germany in German in 48, but there is actually an earlier version which I couldn't find. I could find only the translation, and that's actually the paper we're interested in from 1946, and that's what I gave you to read. So, for the theory and the accompanying experimental work on energy transfer is the most widely known and most influential of all for application. Forster provided an accessible theory in a form that was practical for experimentalists. It is difficult to exaggerate the influence of his work. The extensive widespread use of thread in physics, engineering, chemistry, biology, and medicine are due to the description of energy transfer given by Forster. This is taken from Robert Clegg, The History of Thread, book chapter in Review in Fluorescence. Bob Clegg was a friend. He knew Forster. He's an American scientist who moved to Göttingen, Germany, and worked with Tom Jobin over there for many, many decades. And then in the early 2000s, went back to the US and was in Urbana-Champaign until he died out of brain cancer. And Bob was an expert in thread and wrote many, many fundamental papers. He also wrote this book chapter on the history of thread and gives an analysis on that first paper. So I'm going to use Bob's analysis of the paper. So for those of you who read the paper, What was Forster's motivation to study energy transfer or energy migration? This is how it titled the paper, Energy Migration. Come on, there were 8 or 9 people at least who read the paper. It's in the introduction. What was the motivation? Finding the mechanism for what? What was the problem of that time that he was really trying to answer? It was a big puzzle. Mechanism of assimilation. In other words, you can open the mic if you want. What is assimilation? What does it refer to? No one? Well, it's in the introduction. So we're talking about photosynthesis. There was a big, big puzzle in photosynthesis. People have done some experiments and really could not understand the results. So let's talk about this. Forster apparently became interested in the energy transfer process because of the non-effectiveness of photosynthesis. And Oppenheimer also was dealing with this, although he was also aware of the work of parents. Parents were father and son working in Paris in the early 20th century. They've done all kinds of interesting experiments on quenching, on dipole of molecules studied by fluorescence and how polarization is lost due to energy transfer. But they have not really formulated the problem properly. The experiments were very, very important and later on explained very well by Forster. But basically what they've done all kinds of experiments of fluorescence in cuvette where they look at quenching and polarization effects. Experiments by Emerson and Arnold had shown that the capture and utilization of light energy by plant leaves was much more effective than would be expected if it were required that photons exactly hit the reaction center. That is, there are too few reaction centers in the leaf to explain the very effective extent of energy capture. So basically, in the leaves, you have reaction centers. This is where assimilation occurs. This is basically where you fix carbon. Very relevant to today's, climate crisis. And the leaf is capable of collecting light and putting the energy of that light into those reaction centers. But the number of reaction centers in the leaf is just too little. And people could not understand how come the process is so efficient. So that basically is a question that Forster is trying to answer. So this is taken from his paper. According to measurements by Emerson and Arnold, short-term irradiation with high light intensity, they use sparks. That time, if you wanted a very intense light, you use a carbon electrodes and basically create a spark that generated light, intense light. Produce saturation effects in algae cholera such that the assimilation yield does not exceed a certain threshold, even at arbitrary high light intensities. This limit is reached between 2 and 4 light quanta per 1,000 chlorophyll molecules. The number of light quanta required for the reduction of 1 CO2 molecule. Gaffron and Wall conclude from this that this number of molecules contribute their absorbed energy for the reduction of a single CO2 molecule. As this has to be assumed to take place in a distant location, this interpretation requires energy migration from the chlorophyll molecule to the location where the reduction takes place. So basically, There are many chlorophyll molecules. They are absorbing the light, but there are way too few reaction centers. So how is the energy moving from the chlorophyll to the reaction center if the chlorophylls are far away from the reaction center? So that's a question he's asking. It's really a question about stoichiometry. Previous experiments showed that stoichiometry, that for each reduction of one CO2 molecule, there are 1,000 chlorophylls involved in that process. And somehow those 1,000 chlorophyll molecules move that energy to the reaction center to fix one CO2. Foster knew of these results and reasoned that an extremely efficient transfer of energy between the chlorophyll molecule must be responsible for the eventual diffusion of the energy, which was absorbed over the whole surface of a leaf into the relatively sparse reaction center. So people were familiar with another energy transfer mechanism, which is electron transfer. You're all familiar with electron transfer. It participates in many chemical reactions. There is even photo-induced electron transfer called PET, but the idea of other kind of energy transfer for relatively large distance, that molecules are not in contact, and you can still transfer energy, people had some kind of vague idea, but they did not really understand this. So this is what Foster is working on and trying to solve. He assumed that this energy diffusion is due to energy rapidly hopping between molecules. He was also aware of the earlier work of Perrins and of other data indicating energy transfer over distance longer than the molecular diameter. Again, the idea is to look at energy transfer that is not based on contact between molecules. Nowadays, we have very beautiful structures solved for the light harvesting complexes, the chlorophylls, and the reaction centers. And we can actually do very precise calculations of how energy is transferred from the chlorophylls to the reaction centers. But when Foster worked his theory, obviously he knew nothing. And we'll read a conclusion toward the end of the paper where he predicts exactly this, which was really remarkable. Another example. So basically, these are the chlorophylls. They collect the light and they somehow transfer it to the reaction center. It has to be pointed out that the existence of an assimilation unit and therefore energy migration in the assimilation unit is very controversial. But this time when he writes this paper, there is a debate. Foster then brings other non-biological energy transfer examples, synthesized photochemical reactions, concentration-dependent depolarization and concentration-dependent quenching. These last two refer to the Perrin, the father, and Perrin, the son, and their experiments. And he adds, it is now mostly assumed that in biological processes, the energy migrates without being linked to a material carrier of any kind. So he's setting this apart from electron transfer. There is no actual transfer of material from one place to another. Energy is migrating, and later on, basically the Foster energy transfer is coined or termed as radiation-less energy transfer. We'll talk about this. So what is it really? Think about a radio station transmitting into the air electromagnetic waves, and at home you have an antenna, and you receive these electromagnetic waves translated into voltage signal that goes to your receiver that knows how to decode it into sound waves. So it's a dipole-dipole interaction that is responsible for this energy transfer. You know that in a radio station, you have to have strong enough transducer with large enough power to send those informations to far distances at good signal to noise. So basically, you somehow move by dipole-dipole interaction this energy in the form of electromagnetic waves. When we go down to the molecular description, it's very similar, except that we now work with atoms and molecules, and we have to take into account quantum mechanics. But basically, it's an electromagnetic dipole-dipole interaction between molecules that allows us to have this radiationless energy transfer. So here is a description of the concept. We have, can you see my cursor? Yes? No?
Yes.
Yes, okay. So we always talk about a donor-acceptor pair in terms of energy transfer. Those are two fluorescence molecules, fluorophore dyes, whatever you want to call them, chromophores. You can see here the ground state and the excited state. S0 is the ground state, and D is the first excited state where you have many S1, S2, and so on. You have vibronic manifold in each one of them. If you excite by laser or by lamp, the molecule, it will relax into the ground state by emitting a photon or by generating vibrations and phonons. So you can have radiative recombination or non-radiative recombination. And you have to account for both processes. So basically the quantum yield of a fluorescent molecule depends on how much of the excited energy is relaxed to the ground state through emission of photons versus how much through emission of phonons. Obviously, a good high quantum yield dye means that there is very little waste of energy in terms of vibrations. Most of the energy goes back in forms of photons. The acceptor molecule is the same as the donor molecule. It's another fluorescent molecule, but its energy spacing between the ground state and excited state is slightly smaller than for the donor molecule to obey something we call the resonance condition. So when you take two such molecules, donor and acceptor, one next to each other, where the emission of the donor coincides with the absorption of the acceptor, that's a resonance condition. And they are close enough together. When you excite the molecule, you have free induction decay. You have basically generation of electromagnetic waves, as long as this excitation is coherent before it loses its coherency. And that basically can excite the molecule here without actually providing light at the right frequency. We don't have a laser that excites the acceptor. Just by putting the acceptor closer to the donor is in an excited state. It radiates energy, but it doesn't radiate energy. It generates electromagnetic dipole, and that electromagnetic dipole can excite this acceptor molecule. In terms of the quantum mechanical processes that happen here, we have the pair of donor and acceptor and addition of photon at the energy of the donor excitation that can create an excited donor. This is the asterisk here, plus the unexcited acceptor. And here you can either go to a relaxed state, both donor and acceptor in the ground state, with the emission of vibronic excitations of phonons, or you can relax to the ground state with the emission of photon. So you get the acceptor and donor in the ground state plus a photon emitted from the donor. Alternatively, this excited pair transfer the excitation from the donor to the acceptor. And the acceptor can undergo these two processes as well, either creating vibrations and going to the ground state or emitting a photon in the acceptor frequency and going to the ground state. So these are basically the processes. You can write rate equations, solve the differential equations, and so on. If you do that, you come to what is known as the Forster formula, which says the energy transfer efficiency, how much of that excited energy goes to the acceptor, depends on this KT, that's a rate going from excited donor to excited acceptor, divided by sum of all the rates. KDI plus KD, and that is, once you work out all the equations, can be written as 1 over 1 plus distance divided by some kind of a parameter, which we call R0 or Forster radius, to the 6th power. This is a very, very strong dependency on distance. You can also write it in terms of rates. The rate of FRET is 1 over the fluorescence unperturbed donor fluorescence lifetime times this parameter R0 divided by the distance to the 6th power. So again, this is what we call nowadays the Jablonski diagram. Blue is the excitation of the donor, green is the emission of the donor, and that emission of the donor can excite the acceptor. You have also non-radiative rate, and you have the FRET. So if you excite the acceptor, you can get emission of a red photon, and you can get some vibrations here as well. So this distance dependence of the energy transfer, if you plot it, has a sigmoid shape with a very, very fast dependent on distance around this parameter, which we call R0. R0 is a distance at which the energy transfer efficiency is 50%. So if we put here 1/2 R equals R0, we get this point, which is at the middle of this change from 0 to 100%. 50% is the distance for R0. And for different donor-acceptor dye pairs, we get different R0s. But you can see that along this range, we have almost linear dependence of energy transfer as function of distance. And this is really good news because we can use it as a ruler to measure distances. So here you see some simulations for different R0s. You can see the sigmoid can get very soft if you have a very large R0 or very sharp if you have short R0. Depending on the problem that we have, we'll talk about applications next week when I'll describe you what we are doing with FRETs. But basically, you can use it to measure distances and more importantly, distance changes on macromolecular machines and basically look at proteins in action. So you need to choose the right ruler for whatever the problem is if you're interested in 2, 3, 5 nanometers. So you have to match the R0 to what you want to study. The parameters that make this parameter R0 are spectroscopic and quantum mechanical description of the molecule. J is the spectral overlap between donor and acceptor. Phi D is the quantum efficiency of the donor, and index of refraction, and kappa is something we call the orientational factor. The spectral overlap, here you see absorption in dotted lines and emission in solid line for donor and acceptor. And basically what we mean by this spectral overlap is this hatched area that is the overlap between the emission of the donor and absorption of the acceptor. And basically this integral here gives us this area. The orientational factor is basically we are dealing with two dipoles, dipole for donor and dipole for acceptor. And depending on their relative orientation, you can have more energy transfer or less energy transfer. This is a very notorious parameter because depending on details of your molecular systems, if the molecules are free to rotate, you are in a good shape because you can average and you find out that if you do the right integrals, kappa squared averages to the value of 2/3 if those two dipoles are totally freely rotating. However, if those dyes are sticking to the surface of your macromolecule and there is a fixed permanent angle between the two, the energy transfer can go all the way from 0 to 4. You can imagine that if the two dipoles are parallel to each other, you have maximum energy transfer. In this fashion, if they are parallel in this fashion, you have 1. And if they are totally orthogonal to each other, there is no energy transfer at all. But in most cases, we really refer to the 2/3. We assume that the dyes are freely rotating. There are several ways to measure FRETs. One is based on just the intensity of the donor. So you prepare two samples. You make a sample with donor only and sample with donor and acceptor, and you look at the difference. This is given here, the ratio between the two differences. That's one way to evaluate experimentally the FRET value. Another way is through the excited state lifetime of the donor. You do what is called the time correlated single photon counting measurement of the lifetime of the donor with and without the acceptor. And from that, you can get the FRET efficiency. Or you measure the full spectrum of the pair with and without the acceptor, and basically you write out this expression to calculate the FRET efficiency. So Forster concludes, however, conditions are conceivable which even in high concentration do not allow associates to be present or to be formed after excitation. It would only be required that each chlorophyll molecule is sufficiently deeply embedded in a corresponding protein or that it's bound at a defined position of a protein structure. However, a quantitative treatment of such model on the basis of the theory developed here appears not yet possible for want of sufficient data. So here he makes this amazing prediction in 1946. The first crystal structure of a light harvesting complex is, I don't remember, around 2010 or something like this. So 50 years after this prediction, basically, we have those beautiful structures that I've shown you before. A final clarification of the mechanism we are dealing with here will probably only be possible on the basis of a more complete knowledge of the physiochemical structure of the basic matter involved in it. We only wanted to demonstrate that this type of energy migration in non-biological systems is more frequent than generally assumed, and that by and large, it is a phenomenon which is theoretically understood. We now have an overview of the conditions for its occurrence. Where these are fulfilled in biological systems, energy migration has, of course, to be taken into account for interpretation of the observed effect of light. So to summarize, first, the theory is a basis for our present understanding of threat and most of its applications. Within the validity of his model, which encompasses most of the applications, Forster's original theory still applies. His theory has been tested thoroughly, and the conditions for which his derivation is applicable are met. His theory has always been found to be valid. Forster's original theoretical description of energy sets the stage for all subsequent application of threat in many fields of research, from pure physics to biology, and it is his theory that still is used to interpret experimental results. He extends the original idea of pairings involving the well-known interaction of molecular dipoles. However, his insight and great contribution was to provide the quantitatively correct and very practical description of threat process in terms of experimentally accessible parameters. Threat has been shown to be broadly applicable and extremely informative in determining molecular interactions and to measure molecular distances that are impossible to determine otherwise. All right, so I'll stop sharing the screen now and just give you a heads up for next time. A big part of my own research has to do with threat, but a very unique implementation of threat that is single molecule threat. And we'll discuss the history of ensemble threat versus single molecule threat and many, many applications. So that's the two papers I wanted to analyze and discuss in terms of how to write a good paper. The Watson Crick paper is one page, very simple to read, very, very readable. The Forster paper is a very, very good paper, but much harder to read. And the question, and we asked already that question, how can you have large impact with your results? And how the scientific writing itself affects the impact of whatever the great scientific result that you obtained. The way you write your paper could further increase or decrease the impact. What we'll do next week, I'll give you my story of single molecule threat. We'll look into one of our papers, and you will get basically homework assignment on this paper. I know that most of you have nothing to do with biology. It will be mostly Greek for you, but that's part of what we are doing in this course. We will ask you to read a paper from another field and try to answer questions. So there will be a homework assignment with a date to submit your work. If we have time, we'll also go to answers for that exam, but I'm not sure we'll have time. Any questions, requests, complaints?
First of all, thank you. I just wanted to ask, I saw in the INBAR that it's written that we will have exams, but you said that we won't. So if you can delete it or something, I don't know.
Sorry, I did not understand. Can you repeat?
I just saw in the INBAR, you know, the INBAR, it's like the Moodle, but different.
No, I work with Moodle. I'm an old fart.
Okay, so it's written that we have exams in this course, but you said we don't.
No,
so
just go. Can you go into Moodle?
I don't see that in the Moodle.
Why? It's in there.
No.
It's important.
No,
No,
I just said the exams, we can't see them in the Moodle, but we can see that in the
just
exam
ignore.
schedule.
Okay, just ignore
Okay.
in bar. It's not important.
Okay, thank
This
you.
course is very unofficial. Okay. We require your attendance. We require you to submit an answer to this homework. And then we require you to prepare a PowerPoint presentation, give it to the whole group, and summarize this PowerPoint into 2 pages of a PDF and submit this PDF to me. Those are the requirements for the course. I'm going to give a final grade, and the grade is pass or no pass. That's it. And frankly, you will have to work really hard to fail this course. Okay, so forget about the formalities. I don't know what INVALI is, and I don't really care about it. Okay, so talk to me, and you know, just do what you are asked to do, and you'll be doing fine. Okay, any other questions, requests?
Yeah, so it's a binary grade, right?
Sorry, again?
You said it's binary grade?
Yes. עבר נכשל.
Okay.
More?
Yeah, I got it. Thank you.
Okay, if no more questions, I'm going to post today's slides. I'm going to post today's recording of the meeting. I apologize for not doing this for last week. And we'll see you next Tuesday. So good night, everybody.
Thank you. Good night.
Bye.
Good night, thank you.
Bye.